Good afternoon. Thank you for the invitation to moderate and speak at this session. So I'll be getting creative with biochemistry and molecular biology education. My name is Martin Hicks from Monmouth University, and I'll be joined by Karen Resendes from Westminster College, Gretchen from University of Colorado at Boulder, and Cheryl Clausen-Kuzina at St. Leo University, and Kenneth Hollenbach at Reimagine Science at Genentech. And as they begin their talk, I'll allow them to introduce themselves and give their own background. So today I'll be talking about active drawing of genetics mechanisms and as an undergraduate learning tool. So a lot of this was put together by my students who actually are teaching assistants, and they run review sessions and other sessions using Instagram to get students involved with drawing. So there really is a need for effective teaching method for complex genetic concepts to students. And the challenge is to really integrate drawing and diagramming of abstract mechanisms into the classroom. And this isn't always done with PowerPoints. And the approach we use is active learning to cultivate student success in the genetics course. So what I've done is I've taken basic concepts from the Genetic Society of America, where I focus on gene expression and regulation for one aspect, the nature of genetics material, how genetics variation exists amongst or within species, those patterns of inheritance, and connecting that to molecular biology of gene function. So the structure of the lecture is to do an active stepwise drawing and discussion of concepts in the classroom, where we address a mechanism, usually using a disease process as an example, so students can connect to that, something in a disease that's relatable in the modern world. And then we use reinforcement through recitation or review sessions is what we actually end up calling them, and group collaboration sessions where we encourage students to work together. And the assessment that actually reflects conceptual skills of the students. So that's rather than multiple choice, are we able to move towards drawing these mechanisms as an assessment? The overall evaluation is to increase understanding of molecular genetics topics and concepts, and most importantly, a way to retain these concepts, and then how to apply those in future studies and careers. So the pedagogical design is a collaborative process where we promote active learning in and out of the classroom. And our goal is to have a successful application of student learning through hand drawing. And as you can see here, this is an example of the learning environment where I'm drawing up here. We have students on their whiteboards. We have students sometimes set these whiteboards up on the glass wall. And I usually rotate students on the front board also, where they come in and do their drawing. So the goal is to have the students come in and begin actively drawing and discussing the material. So they come in preemptively already going through the learning process, and then they come into the classroom and test those out. So the strategies such as comparison and reductionism are often used. And when I say comparison among student drawings and my drawings, and then a reductionism where we go from the macro, the large picture, into the molecular. And this really seems to help with student understanding. So my hypothesis in all of this is, can active hand drawing of complex molecular biology topics in lecture, group recitation, and instructor-led collaborations increase student learning in undergraduate genetics and molecular biology lecture and laboratory courses? So I'm going to give an example of the progression of metaphase chromosome to the structure of bDNA. So on the right, you have a drawing that we can excerpt from a textbook, for example. So we evaluate this printed drawing to build a base of knowledge or of a concept. We then generate a relatable hand-drawn image. We do a theoretical macroscopic, the big picture, to the molecular, what's happening at a molecular level, and even atomic, atomic sometimes, electronegativity, for example. And we label and diagram using colors to distinguish structures. So the overall evaluation that we're doing is setting a foundation for understanding DNA, histone, octamer structure, and epigenetics. So this is our rendering from the textbook. Then we take that and we make a workable drawing showing the microscopic or macroscopic to molecular progression from metaphase. So here we have the metaphase chromosome demonstrating the nanometers and how we can actually go into chromatin structure and loop domains, and then what we call a solenoid. That solenoid's used differently for different people, but we have 6 nucleosomes surrounding together form a solenoid of about 30 nanometers. Then going into about 10 nanometers, where we have H3 and H4 histones as a tetramer, and H2 and HB histones as a tetramer, forming the octamer. Now, if we take that out, 147 base pairs, we can have that bDNA, which is 2 nanometers across. So this is a way for students to draw and visualize the breakdown of the metaphase chromosome to bDNA. And really, there's a linear progression to this. So then we even move more. What is actually happening in that histone? So we have histone 2A, 2B, 3, and 4. And we show how there's a dimer formation, a tetramer, and how this histone octamer has lysine residues which have affinity for the phosphates of DNA. So students actually begin to, I can say this, and then the students begin to draw and simulate what I'm saying verbally often. So for example, here we have histone acetylation where we have the lysine residues. Here we have the amine, which can generally have a positive charge, and that can interact with DNA. So here we have a histone acetyltransferases where that is coated with an acetyl group, and then that can go back to form the amine group. So this is a way that the actual lysine residue becomes or loses affinity for the DNA. So students actually now my students have actually started drawing right here where my mouse is. They've drawn the phosphates there, and then they show how the phosphate now of the DNA is not connected in this one, for example. So it really does help students visualize what's happening from a macro to micro level. Now, we can also take the example of double-strand break repair. So again, here we have an evaluated printed drawing to build a base knowledge of a concept. Now those students generate a relatable hand-drawn image, and then from this, we've actually done a comparative strategy analyzing double-strand breaks from ionizing radiation, and then how that can be related to SPO2 mediated double-strand breaks during meiosis in prophase 1. So here we have the labeling drawing using colors, and then a comprehension of DNA repair and connecting that to genetic variation from during strand invasion. So here we have a student rendering of that same image where we have a double-strand break repair from X-rays, gamma rays, cosmic rays. Here we have the break with 5' recessed ends. And in this image, they're showing that the sister chromatid in a mitotic cell is able to be a template to create an identical copy of that chromosomal region where there was a double-strand break. And students really begin to relate why we are diploid species just doing this. They connect that right away. And then they see how that's repaired. And now, since students understand this, when they learn about meiosis, that SPO2, that the actual meiosis, the meiotic process, is likely evolved from double-strand breaks, and they relate this to a SPO2 mediated, where we actually have an enzyme that makes double-strand breaks in the germline cells during prophase 1. That keeps on moving. So SPO2 enzyme leads to double-strand breaks, and in this case, we have the double-strand breaks. We have the 5' recessed ends, but instead of that sister chromatid, which is identical, we can actually have a homologous chromosome. We can have the maternal copy being the template for building and filling in those gaps. And in that way, we're not only repairing that break, we're bringing in another version of that same genetic region, and this leads to genetic diversity. And the students really, in this example, connect why we are diploid to connect how diversity occurs in meiotic cells. And I actually learned so much just by us drawing this. If we never drew this, I myself wouldn't understand this connection as well either. So through this drawing, we've actually advanced learning, and students carry this forth in their future careers. So I'd like to have this last example where we look at the Mendelian genetics of the ABO blood group. So here I'm skipping the evaluation of a printed or word generated version of a blood type. But here we have, for example, a blood type where we do a chi-squared. So this would actually be the fucose gene, where we have a Mendelian inheritance where we have homozygous dominant, heterozygous dominant, and we have the recessive fucose gene. And then here we have the AB blood type. So students can do a dihybrid cross with that using a fork line method and develop the ratios. Now we can connect what's happening at a Mendelian inheritance level to what's happening at a molecular biology level. So we take that ABO blood group, understanding the role of the H precursor. So here we have the FUT1 gene bringing in fucose. So we have galactose and acetylglucosamine, which is a disaccharide becoming a trisaccharide with the fucose. And then the students see that the A allele will actually put on the acetylgalactosamine onto the galactose here, or the B allele will put on the galactose at the same corresponding region on the galactose, forming the B antigen. So what happens in this drawing is students begin to connect the Mendelian genetics of the ABO blood group to the molecular biology, and they're able to, through this drawing, even relate the epistasis, how this upstream gene can determine the fate of this sugar as it becomes either the H substrate type O, the A antigen type A, or the B antigen below. So this drawing is really connected a concept that is often abstract in genetics and now becomes, they're able to retain it for a lifetime, I believe. So in these student learning outcomes, the goal is to create new illustrations that are student generated to cultivate deeper understanding of genetics lecture concepts and student analysis and connection of topics. So students actually bring a more dynamic version than I do. They all draw better than I do. And I believe they improve retention and they increase their mastery of the material for future studies. And here I have just, all right, here I have the e-learning version of that from 2018 of how we really want to kind of incorporate learning. So the current directions are measurement of student learning success through inventory assessments. And I think some of my colleagues here might have some ways to help me with that, which I'm currently doing, but I always am open to new ways. A continued use of review sessions as positive reinforcement, collaboration with other professionals to promote active learning in collegiate science courses. So my school, I have 25 students in this course, but I believe that we can generate artificial intelligence to read student drawings to begin to see if we can grade these in a more dynamic way where we're not actually doing one by one. And this can replace potentially our multiple choice questions, which I think are getting away from actual learning. And hopefully we can do, I'm publishing a peer review and publish these drawings as a learning tool in the future. So if you have any questions, please send them my way, and I look forward to having discussions with all of you. Thank you so much.
Thank you, Martin. What a really interesting talk. I'll step in to ask you questions. Seems like a chair shouldn't have to ask himself all their own questions. So just a reminder for everyone watching, if you do have questions, please feel free to just write those in the ask a question spot on the web as they come to you. No need to wait till the end of the talk. But Martin, I'll start. You kind of hinted there at the end about digital or machine learning ways to handle these drawings. Is everything you're doing is by hand now, but are you moving to a digital format? Is that the plan?
So what I do is the students actually, especially with the Zoom, what the students do is they all have their whiteboards. They use Adobe Scan and they take a picture of each drawing and they convert it into a single PDF. And they deposit that onto our Dropboxes, and then they're easily evaluated. And I actually just scan those myself with my own eyes, and then I really do the further value. I evaluate them more strictly on the exams and tests. But I do believe that we are accumulating these drawings that then could be put into a system of artificial intelligence where they actually begin to figure out what is correct, what's not correct, and develop a framework for grading these without the time that it takes. For me, I have 25 students, I can do it. But in larger courses, if we want to expand this learning and facilitate this type of learning and testing, I think it would require a system that is based on artificial intelligence and the human eye after that. Does that answer your question, Ken?
Yeah, thank you. I have another question coming in about just academic honesty and the kind of the challenge that this directly addresses. It's hard to fake a drawing. We all have, I'm a terrible drawer. I'm sure you'd know right away it was me. Have you seen any benefits along those lines?
Well, you bring up something. We actually had a department meeting yesterday where people were talking about how do we address this, especially with the online courses. And I think we have to design, you can't cheat. It's a trick. If they can actually do the linear drawing there, they got it. So, and I look on the Chegg and other cheating places, they told me that everybody should look yesterday at a department meeting, and there's nothing there because it doesn't help. What helps, and if there was something there, it would actually help students to learn. Do you know what?
Yeah.
You can't fake it. It's by concept, you can't fake. You learn it. And if there's a gap in the mechanism, you're going to see it. So I've actually filled in my gaps as a teacher. Sometimes we kind of sidestep the things we don't know, and I don't do that anymore. And I'm comfortable with that. I'm comfortable filling in those gaps, and that's a blessing. And my same gaps were I can actually anticipate where the students are going to have gaps because I've started to uncover my own gaps. But and then I allow the students to go through that process in their drawing.
Awesome, thank you. We have a couple other questions, but I think for time we should probably move on. So Martin, I'll hand it back to you.
All right, thank you so much. Just one second. So our next speaker is Karen Resendes, and she's going to be talking about using student-generated MEMS to review content and build class community. So Karen, if you want to share your screen. And
I am sharing my screen now.
And introduce yourself.
Hi, everyone. So yes, I'm Karen Resendes. I'm an associate professor of biology at Westminster College outside of Pittsburgh, PA. And today I'm going to talk to you about the use of memes in my classroom, which kind of started pretty serendipitously last spring in one of my courses, spring of 2020. And so as I kind of alluded to, memes are really not something I ever thought that I would use in my personal pedagogy. It wouldn't really have not occurred to me that this was something that students could use to learn. And so if we think about first, what is a meme? And so the kind of general definition you can find on the internet, right, is that it's something amusing or interesting, often a captioned picture or video that you spread online, right, often through social media. And so the way this started in my classroom is that one student, Tyler Snodgrass, at the end of class, sent an email, replied all to an email I had sent, which was probably something important about the class, and shared the meme that you see on your screen right now. So we had just learned a particular concept. This is a cancer biology course that I teach. So you can see Tyler's meme here, and you can see Annika and other students' description of that meme. That it's about AKT, which is a molecule that phosphorylates P21 and sequesters it in the cytoplasm. And typically it would be entering the nucleus, but AKT when phosphorylating it is, quote, keeping it out of the swamp, keeping it out of the nucleus. And so we have meme, we have Shrek here, keep out of the swamp. okay? This started a just snowball effect of memes in my class. And so the first piece of this were that the students started to email each other back and forth and kept me on this thread. Every time after class, sometimes during class, they were sending memes on the concepts that we had just learned. And so I took what they kind of bubbled up and made it part of the class. And so that email thread was the first part of it. But then on the first exam of the semester, Tyler's meme showed up. Use this meme to explain how AKT and P21 work together. And so I used it both that way as exam questions. And then when the course transitioned online, because this was spring 2020, they were taking their exams online, so they could, as their last test question, drop in a meme they had created as a bonus answer. And so I'm at a smaller school, so there's 10 students in this class. 60% of those students went on to continuously create their own memes without any prompting. Some of them said they only made maybe 3. There were a lot of them that said more 9 or 10 throughout the semester. But they said they all continued to look at the memes that each other made, right? So I have another example here. We're looking at DNA replication and how a microsatellite is going to make that much tougher, and we're going to stutter. And we see Nina is explaining down at the bottom what Natalie has created for us. And that's a big part of where this started to come into play in learning. I became interested in how this was helping the students with the content, as opposed to just being kind of some fun little thing that we were doing. And so at the end of the course, I had the students take a short survey about their experience. And we talked first about whether it was helping them to learn concepts. And so we have a quote here from Natalie, who made this meme. And she said, I had a really hard time learning the section about DNA damage repair and remembering how it fit together before I started building my memes. And so here we have her description of her personal meme, right? That an alkylating agent is trying to alkylate the DNA, and MGMT just punching right through and taking those off, taking those away. And so when the students were asked, about 66% said that creating memes helped them learn, and that's comparable with the number of students who are doing this on their own. 88% really agreed that viewing someone else's meme helped them to learn something. And what I think is interesting here is that's about what they ranked just taking notes. So making these memes or looking at people's memes are kind of helping them the same way, because they're writing something down and helping to learn that concept. I similarly asked them about this idea of using the memes to learn, to kind of review, go back over what you're doing. So I have that same meme here, but now I have another student's description of the meme. And you can see it's pretty similar to what she described herself. And so she's going back and reviewing the content by looking at her classmates' meme. And we can see here that they're agreeing that creating memes help them to review, viewing others help them to review. And when they compare that to just going back over their notes, it's relatively similar. And when I compare that to something that is more active, so drawing, going back and making diagrams like we just talked about in the previous talk, right? This is also kind of helping in a similar way to kind of synthesize the information. I think the thing, though, that really surprised me about this was the way it helped build a class community. And so I said this is a small class, only about 10 students, but they're all from very different majors. And so they aren't necessarily students who would commonly connect with one another. And so the students really felt that this helped kind of build a community in the classroom. They were more willing to talk to each other and get to know each other, and they connected with each other outside of the classroom. And this really helped too. Like I said, this is last spring. So we went home for spring break and never came back. And so the meme thread was kind of a point of sanity and connection for a lot of these students when we went home. And so they helped it kind of bolster and maintain that community. The other community part is the memes weren't always about the content. So my students hate EMSAs with a passion. And so they are burning them with SpongeBob, and they are quite happy to do that. And if I look at when they describe it, they all describe this is just the way we think about it. This second button pushing meme comes up a lot when they're doing things on community. Again, this is right after we went home, and everybody is very concerned and panicked about what's going to happen with this pandemic, after having kind of transitioned out of learning every week how there's so many mutations and changes that can cause cancer. So they're connecting those two. And again, it's a way for them to connect with one another. And so because it had kind of bubbled up in this class and they all seemed to love it, I thought, let me try putting this in one of my other classes in the fall. And so I was a little bit nervous in that because it was going to be my idea that we do memes and not their idea of building up. But oh my gosh, they ran with it. And so because I was bringing it into the classroom for the first time, I kept it as bonus. So there was, after every unit, give me a meme for the content. So this is a cell biology class. So there's a couple cell cycle and signaling things that you're seeing here. It was extra credit. You can only get the same amount of points for 1 or like 10, but I still got 10 from certain students. They were just that into the idea. And then we turned it into a competition. Blinded, you get them all. You vote for your favorite ones. They get a little bit more bonus if they win. The students who were in my cancer class in the spring, when I told them this was happening, they wanted the link. They were still so into it that they were voting on the current students' memes. So we have Morgan's meme here on cell cycle. Noah, who I think is out there listening right now, has this great meme about how Raph is so excited to phosphorylate MEC, the next thing in the pathway. But then I saw the same thing in this course too. Just as many of the memes were about the experience. And again, this is a class, there's about 25 students in this class, but from very different majors. We are socially distanced, 20 kids in a massive lecture hall. So it's a way for them to connect. I put a lot of these up, especially these ones down at the bottom, before class to see what they, what people were saying. There's so many acronyms, there's so many proteins. Oh my God, don't tell me another one, Dr. Resendes. I like to use lots of cheesy analogies in my class. And last semester, everything was a bust for some reason. I can't explain why, but that is, the joke here on that second one. And so again, it's a way to get them communicating and talking to each other, someone else that they can connect to in the class. And so kind of what it comes down to now is, this seems to be something they like doing. How do we turn this into something that is a more definitive learning experience? So when I asked the students, how should I go about doing this? They suggest that I bring some of my memes into lecture. Mine are horrible. And so I think that may not be the way to do it, or I may cycle back previous students' ones. But they've suggested exercises like a fill in the blank, provide the meme, they have to build the story, or flashcards. They still like the idea of the memes on the test, even though they got mad when they got points off because they didn't quite explain the meme right. They still think that was a good idea. And you're going to see the next couple slides. I'm not going to go through every meme, but I could not put a meme on every slide. So it may or may not be your thing, but you can see we've got the cell cycle restriction point again on this one. The top one is about separating chromosomes in mitosis. And I wanted to point out that I'm probably not the only one doing this, but it's not really out there in the literature right now. If you look at what's out there, the top two studies here, Mendez, Reguera, and Brown, both came out and this past year, and they were about the same kind of thing, where it just started to be something they could do when their classes went online to help people communicate with one another. This is a med school class and a pharmacy class, but you're seeing it kind of similarly used. Discussion boards, optional, informal, but it's building community. We're using contests. But then I really like this idea that Brown brings up about it's a way to apply and reinforce concepts. And something I hadn't really thought about, which is to identify misconceptions and gaps. So if your meme doesn't quite make sense, then maybe we need to go back over that concept. And you can see that again in the third study that I found here, Underwood and Caro. This was a couple years of using an exercise in a undergraduate chemistry course. And what they're doing is a review session, similar to what I kind of just suggested. You give students a meme, they work in individual groups, and they come up with whatever concept they want to apply it. And then they share and discuss with one another. So then they have to explain why their meme is funny. They have to explain why that applies to the concept. And again, you can identify misconceptions in the content here. Okay, and so on my last slide, I just wanted to acknowledge the students who made this a thing. So Tyler Snodgrass, who started this all in spring 2020, and all the students in my cancer biology course, and the students in my cell and molecular course this past fall, who kind of took this as a new thing and totally ran with it.
Great talk, Karen. Can you guys hear me? So that great talk, and I really connected with methylguanosine methyltransferase. I actually have my students drawing the acylation of guanosine. I think there's a way that we can connect these to the visual even. Is there a link that you could help us start to participate in? I would love for my students here to participate in something like that. Can we start doing something like that if you don't have it?
Yeah, I'd love to, because I love anything active where you're adding things in. So drawing is a big thing for me, and this is the same way of doing it. And so maybe it's building that bigger community. You know, I put that out for a broader vote and have your students vote on my students' memes, and my students can look at your drawings and see if they can understand the pathway. I love that.
I think we have to do something like this. And then how are you going to, so the literature, how are you planning to put this out in the literature so we can bring like MEMS into the literature and how they become a teaching tool?
And I think it's kind of tough because I'm torn between the few that are out there are more these kind of case studies. Here's what I did, and the students seem to like it. I'd love to take this to the next step and bring some education-based research into it on actually looking at, you know, does it help them remember or revise their concepts? So I'm a big, deeper education research type person, and so I have to kind of think about. I thought I'd do that this year with the fall course, and I should have known that this year you can't get anything accomplished. So that is one of my goals, is to try and think about how do I turn this into an actual research study on how doing these helps the students to apply and reinforce concepts.
So do we have any other questions out there? I think we're at time, Karen. So it would be great if you could send your communication. We can build a collaboration where we're working to drawing MEMS. We can actually create a community where students are making it popular, right? Making these topics popular. All right. So our next speaker is Gretchen from University of Colorado, Boulder. Would you be kind enough to introduce yourself and share your research? Gretchen, and then over to you.
Yeah, thank you, Martin. I'm going to share my screen real quick. All right. Yeah, and as he said, I'm Gretchen Hoyer from the University of Colorado, Boulder, Department of Molecular, Cellular, and Developmental Biology. And today I'm going to be talking about me and my co-authors' research on collaborative online concept mapping for complex gene expression studies and undergraduate research-based courses. So I'm going to give a quick outline of what we're going to be going through today. First, I'm going to talk about the background and need for this research. I'm going to talk about the course-based undergraduate research experience where we implemented this concept mapping software. It's called the Python project. I'm going to talk about what concept mapping is and how it could enhance cures like the Python project. Next, I'm going to talk about the methods that we used here. So there were 2 semesters of implementation of the concept map. We had a semester with full implementation and a semester with partial implementation. And then we looked at the discussion sections for the final research papers and came up with a way to code and see whether the students' scientific writing improved when they got to use the concept map. And then I'm going to go through the results. We're going to talk about what the student perceptions were of the software, how much they interacted with the concept map, and also we're going to talk about those coding results that I discussed previously. And finally, just go through what are the implications, what is the future of concept mapping in cures like the Python project. So a little bit of background on the class. Like I said before, the Python project is an upper division course-based undergraduate research experience, or CURE. CURES allow students to create their own unique research projects and engage in independent research in the context of a course. So this is a little bit more accessible than traditional apprentice-based experiences. In the Python project, students perform a gene expression study on a single gene involved in the conversion of cholesterol to bile acid in Burmese pythons. And this research may have applications for human fatty acid liver disease. And at the end of the semester, all the students produce a manuscript style research paper, which was the center of a lot of our analysis. So a little bit of the need for this project. Students in this course typically show a pretty poor understanding of the pathway their gene of interest is involved in and the way that the different genes interact with each other. So the question we asked here was, will the use of concept mapping software increase the complexity and depth of students' scientific writing in the Python project? And our hypothesis was that their generation and engagement with an interactive online concept map that summarizes both current knowledge in the field and the addition of their gene of interest, the gene that they're studying, will enhance student understanding of these genetic relationships. So a little bit of background on concept maps. You can see a diagram of one here. It's a way to organize information in a hierarchy and a schema that you can put a lot of complex information on one page. We specifically used Mindomo software in this semester, and Mindomo is a collaborative online software that allows students to work remotely together, which obviously, as we've seen over the last year and a half, is something that is really useful to have in the toolkit. You can see here pictures that were actually generated during the semester of the concept map. So you can see students can add their gene of interest. They can add stuff about the disease and the pathologies of it. They can kind of put anything on there that they think is useful and have them related to each other in a way that you can see all at the same time. So concept mapping has been studied all the way from elementary school to graduate medical education, but it hasn't been really extensively studied in an undergraduate setting, and especially has not been studied in courses or lab courses like the Python project. But it's been seen to be beneficial when measured by student test performance. It's seen increased knowledge retention and a self-reported reduction in anxiety, an increase in satisfaction and confidence. So I'm going to go quickly through the methods that we used in this study. We had 62 study participants total. These ranged across 4 semesters and 3 interventions. There was a full implementation semester, and I'll discuss what all this means in a second. We had 16 students, partial implementation semester with 18 students, and control was 28 total across 2 semesters. And this was done on purpose just to control for the quality of class over time. So if we look over here at this figure, I'm going to run through kind of what that looked like for each implementation. First of all, across all 3 semesters, they had identical curriculum, and all of the final research papers discussion sections were scored with a scoring system I'll run through later. And if we look at full implementation here, they received a tutorial and they created a concept map from scratch. So using Mindomo, they went in, added all of the stuff that they thought would be important, added their gene of interest, other genes in the pathway, other pathways the gene product might be involved in, and then they edited this down to something that they were happy with. At the end of the semester, instead of the traditional presentation of a research poster, students presented the concept map orally to members of the department. They were also mentored in class on concept map usage. On the other hand, for the partial implementation semester, they still received the same tutorial, but instead of creating the concept map from scratch, they edited the existing concept map that the full implementation students had created before. So it was a way to see whether the creation of the concept map is the important part here, or it's more like just interacting with the concept map in general. Instead of presenting the concept map orally, they presented the individual research posters at the end of the semester. Something to note here as well, the students spent the same amount of time, no matter what semester it was, kind of thinking about their gene in the context of the pathway and in the context of other genes. But in the partial implementation semester, they were just supplemented with other materials instead of the Mindomo concept map. So I'm going to run through kind of the analysis that we used here and the coding system that we came up with to look at the discussion sections. So there was no validated measure for this type of analysis for looking at complexity and depth of student scientific writing. So we developed one from other sources, and we came up with 2 codes, a complexity score and a depth score. The complexity score ranges from 0 to 5, and we're looking at whether students can acknowledge the roles of other genes studied in the course in relation to their gene of interest. And then we looked at a depth score that ranges from 0 to 4, and we saw whether students could discuss the overall pathway that their gene is involved in and potentially other pathways that the gene's product might interact with. The analysis of the assigning of codes was done by 2 independent evaluators, and then any discrepancies were resolved jointly. And then we calculated the averages of these codes and did a comparison of means with a p-value set to less than 0.05. I'm going to run very quickly through what the scoring kind of looked like. So again, the complexity score is looking at multiple different genes interacting with each other. It ranges from 0, which meant they only discussed their gene of interest. They didn't mention any other genes at all, to a score of 5, which means they mentioned and discussed 5 plus additional genes in addition to their gene of interest. Same for the depth score. Again, we're talking about the pathway and the mechanism that the gene of interest is involved in. It goes from a score of 0, which meant they didn't discuss the pathway or mechanism at all, to a score of 4, which meant that they discussed the role of their gene of interest in all pathways and mechanisms that were mentioned in this discussion section. So here are some quick results. First of all, we surveyed the students and saw what they thought about the software. So something that I want to point out here is that when they were surveyed on their perspective towards concept mapping, it was largely positive. If you look at figure C here, the majority of students selected between somewhat agree and strongly agree. And the light gray bars are full implementation, dark gray bars are partial. To the question that the concept map helped me with my sense of collaboration with other members of the lab. If you look at figure A as well, you can see that they on the whole, felt very strongly that the concept map helped them understand their gene in the context of other genes studied in the class. Also, we asked the students about the ease of use of the software and their time spent using it. So you can see that most students found the concept map somewhat difficult to use or were neutral with regards to ease of use. Time spent with the map over here in figure B did not correlate with the reported level of difficulty with the map. And students spent the same amount of time considering their gene, again, in both semesters. So partial implementation students were supplemented with other sources of ways to kind of think about their gene. And most students in the full implementation semester, again, these light gray bars over here, as you can see, spent 2 to 5 hours on the map, and the partial implementation students spent a good amount less, again, because the partial implementation students didn't have to create the map from scratch, so that saved them some time. We asked students for their recommendation regarding concept mapping and what they thought the perceived value was. You can see here, as far as the recommendation goes, the partial implementation students were a lot happier with the emphasis that was used in the course. They felt like it should be used with the same emphasis as opposed to the full implementation students. And you can see, as far as perceived value, the vast majority found it pretty valuable from both semesters. We also kind of looked at the concept mapped interaction. We saw how much students interacted with the map in both semesters. And so interactions we measured were whether they created nodes, moved nodes, edited nodes in the MyNomo software, and we tallied those up. And we found that the average number of interactions per student was not significantly different between the full and partial implementation semesters, suggesting that even though the time spent might be different, the level of investment is around the same. And so here are the results of that coding analysis that we did on the discussion sections. The top bar here is full implementation, middle bar is partial, bottom bar is control. So the important thing here is that you can see that for both scores, the full implementation semester students' discussion sections were found to have much greater, statistically significantly greater scores for both complexity and depth of their scientific writing when we compared to a control. And for the partial implementation semester, it showed trending but non-significant differences. So you can kind of see from these graphs, as the level of investment with the map and level of interaction with the map went up, it seems like the depth and complexity and understanding of the gene in the pathway in the discussion sections went up as well, which is what we were looking for. So that was great. And I'm going to talk quickly about the implications of this. First of all, concept mapping software is easily adaptable to a remote format. Obviously, this is another thing that's really important to have, as we all had to do remote learning over the last year and a half, something that you can have in your toolkit, whether there's a remote portion to the lab or not. The implementation of concept mapping software was well received by the students. They said it increased their collaboration in lab. They found it very valuable. And then finally, concept map integration increased the students' understanding of the relationship between their gene of interest, other genes, and the overarching gene pathways that the genes were involved in. Some quick acknowledgements. I'd like to acknowledge Dr. Leinwand, who is the PI for the Python project, the CU Department of MCDB, and Emma Burt, who made some early contributions to this project. Now I can answer any questions.
Great talk, Gretchen.
Yeah.
I want to use concept maps myself. I've never. Are you able to share the software or a link to the software that you've been using so that other people can use that?
Yeah, it's just minddomo.com. It was something that was free for the students. I'm not sure exactly how much the university paid for it, but it's just Minddomo concept mapping software online.
Okay, all right, that's great. And I feel like these all connect, like whether it's MEMS, whether it's drawing, and then the concepts map connect. So if I'm doing my drawings, I've had students do layers of going from the molecular to the macro, but I've never been able to figure out a way to integrate those. I'm wondering if during the semester a student could build their concept map as they do their drawings. I'm wondering if this program could help with that.
Yeah, definitely. That was something I was thinking as well when I was hearing both you and Karen presenting your work, that concept mapping is a nice way to kind of put those visual aids that you're using in your course and kind of put them in one big thing that many students can look at and see the relationships between them.
It could be a studying tool. I mean, they can associate the MEM as a kind of mnemonic device with the drawing, and then find it through their concept map.
Yeah, absolutely.
I can see this in my head, but I don't know how to build the connections yet. Any more questions coming? So we have, do we have any questions coming from the other people in the group? Karen, Ken?
Gretchen, I'm curious. Do you think this is a larger concept that like universities will adopt, or like was this just kind of I guess I'm asking, like, how did the administration and the teachers and like the other stakeholders in this process enjoy using this tool other than the students?
Yeah, that's a great question. I don't think that's something that we looked at like super specifically in a research sense, but I think that the majority of people that saw the concept map in the department were like found it a really useful tool, because again, in these upper division courses where there's like a lot of complexity with the genes and how they relate to each other, I think everyone can sometimes benefit from having a visual way to represent really complicated things like that. So I think overall it was really well received in the department, and people were impressed by what the students did.
All right, great job, Gretchen. So we're moving on to the next speaker, Cheryl Clausen-Kozina from St. Leo University. Could you introduce yourself and share a little bit about yourself, Cheryl?
Sure thing. I'll go ahead and share my screen. Okay, so I'm Cheryl Kozina. I'm at St. Leo University, which is a small PUI in Florida. I want to thank the organizers for giving me a chance to speak today. And before I get too far in, I want to make sure I acknowledge my partner in crime, Pam. She is one of our instructors of humanities and arts, so not typically a conference she'd be attending, but she is absolutely behind the scenes very helpful in the whole gamification process. And so I will be talking to you today about gamification of general education science courses, in particular for online delivery. And so I want to start, for people who don't know, talk a little bit about what gamification or gameful learning is. And this is just the use of game design elements in non-gamed contexts. And this can include things like simulations, where you have games modeled after natural or man-made systems, where the players have to achieve some kind of goal, and also strategy, where you can recreate a historical or fictional scenario in which players devise an appropriate strategy to achieve a goal. Now, there's other types of games. I just felt these were the most prevalent in the sciences. And so why use gameful learning in your class? Well, it's been shown that digital games are effective in promoting scientific knowledge and concept learning. And they have been shown to affect student confidence, motivation, and engagement. I will point out, though, that there have been some studies that have shown that students can find games enjoyable, but it won't necessarily affect learning outcomes. So I'll come back to that a little bit. And finally, what I think is most important for this group is that the educational benefits of GBL are particularly apparent in subjects over which students report greater anxiety. And I think anyone in this room who has had to teach non-majors in a science course can agree that they experience a lot of anxiety. They don't want to be there, they're scared of it, they haven't had a science course since high school, if that. And so if games can provide a benefit to these students and help them to have a little bit of less fear, I think that's an important thing. And I want to encourage you that you don't have to reinvent the wheel. So if you're sitting there thinking, I don't have time to write a game, I didn't either. Think about what you're already doing in your face-to-face classes. Do you do active learning already? Can you adapt something that you're doing already to a game? Use outside resources. Course Source, the National Center for Case Study Teaching in Science, I use a lot. I'm a geneticist by training, so I use Teach Genetics quite a bit. And also, as I'm sure many of us here are familiar with, our society websites often will have a section that includes educational resources. So ASBMB obviously does, GSA and ASM do as well. So don't feel like you have to start from nothing and write a game. Use what's already out there and then try to adapt it to online. If you are interested in doing gamification, a couple quick best practices. The game should be supplementary. So this should be something that you do in addition to your normal course activities, your normal lectures, and anything else. Use it as a review, for example, but not necessarily for a grade. So none of my games are actually tied to any assessments. They're meant to kind of bring together everything the students have already learned and help them on those ultimate assessments. It's also important to provide meaningful feedback. Just having the game for game's sake isn't good enough if the students aren't understanding why they're not doing well or why they're missing some of these game elements. So the science course that I gamified was one of our 100 level science courses called Is Evolution True? Your Inner Fish. And you can see the modules that we go through in this course. We have the scientific method, we talk about fossils, molecular biology, and human evolution, for example. This fulfills our Science in a Changing World credit for the students. They have to take 2 of those before they can graduate. And while we do teach this course face-to-face in our 16-week semesters, the focus here was on our 8-week fully online asynchronous sections. Now, when I came into this, the course had been online for several years, but in the summer of 2017, I was tasked with reformatting it and chose gamification as a method to do that. And so this is an example of one of the games that I adapted. So I only have time to really show you one today. But as you can see, I took this from the National Center for Case Study Teaching and Science. Didn't write this myself, but it was using the study of ancient hominin species in a dating game format. So our eligible bachelorette, Lucy, is going to ask her bachelors, Homo erectus, Neanderthal, Homo sapiens, some questions about what their dates might look like. And based on their answers, she can figure out some things about them, like, had they harnessed fire, for example? And so my learning designer was absolutely amazing, and she took this and just ran with it. And so this is what our version looks like online. And when the students initially click into this, there would be kind of 60s music playing. And when they get into the game itself, we have our cartoon bachelorette and her eligible bachelors. And so our learning designers did record voices over this as well, but for accessibility, they also have text they can click on and read through as well. As the students mouse over each of our gentlemen here, it would pop up with their scientific name. So obviously, cartoons aren't really going to be true to form and not really tell them what they need to know about who these bachelors are. So you can see an example for one of them. I like a man who is smart with a good sized brain, so getting at that brain size. I want a man that can work with his hands and will cook me a candlelit dinner, so harnessing fire and tool use. Now the age gets a little weird because we're really just trying to hone in on when they were present on Earth. So 2 million years ago, not his actual age. And here's where we get some big clues for this one. I will also like it when my man is bigger than me, 25% larger, strong silent type. Those are big hints for this gentleman. And caring, willing to care for me if I were sick. Okay, so at this point, hopefully the students are figuring out some of these characteristics and who this is describing, and maybe you are too, if you've had an anthropology class. And so here comes that feedback. If the students chose correctly, Homo erectus, they get this feedback. Yes, he had a big brain, about the same size as our own brain. He used tools and fire, lived 140,000 to 1.8 million years ago, so right in that range that she liked. Males were 25% larger, that's the big hint here. But they didn't have an advanced language, so strong silent type. And also were known to care for their weak or ill. But you can see if the student chose a wrong answer, it does also provide feedback. This bachelor is a little bit too old and can't harness fire, so there will be no candlelit dinner. And his brain might be a little bit small. Okay, so you get the idea of what they would go through here to review for their quiz that's coming up on these different hominin species. And so obviously we're all scientists. The question is, does this work? The first outcome that we looked at was DFW rates, because we're obviously very concerned with this in a non-majors general education course. And so far the DFW rates are steady, but we do have some interesting standout terms. So before the remodel, we had never seen a semester with less than 25% DFW, but we've seen 2 since the reformat. So we're at least moving in the right direction, but this kind of jives with what we saw from Fu et al., that you don't necessarily always see improvements to learning outcomes, but the students may still have a positive view of the game. So we next looked at course evaluations because we wanted to make sure that they were having this positive view. And so the first question we looked at was instructional activities helped me learn the course content. And you can see that we are seeing an improvement in this outcome. We also looked at audio video presentations, helped me understand the course content. Technically, this is not what we're asking about. We're asking about a different part of the course, but in case the students were confused, we glanced at this as well, and they also saw an improvement there. We also looked at the question, I gained knowledge about the course content. I think this is one of the more important ones. When we're talking about general education courses, these students aren't sticking with science. We just want them to get something out of this class, become more science literate, and they are. They're getting something from this class, which is important. I do want to point out, if you're noticing the N values for this, unfortunately, out of my control, in 2016, we changed our course evaluation format. So I can't easily get more than 33 individual sections for the old format without doing some definite data massaging, because that was on a 5-point scale. This is on a 4-point scale, and the questions don't perfectly align. So that is an unfortunate thing that we ran into. Some of the other things that I looked at that did not have an improvement, I learned to analyze and critically evaluate ideas, arguments, and points of view. I learned to apply course concepts to solve problems and make decisions. And so that higher order thinking were not there. And again, this sort of goes along with what we've already seen in the literature, that this is going to help with student satisfaction, but not necessarily with that higher order, deeper thinking. We also looked at the question, the instructor effectively used examples or illustrations, just in case the students had any confusion about instructor versus instructional design. And we didn't see a difference there. And then likewise, the course materials helped me learn that question, again, shouldn't be applied to the gamification. That should mean things like their textbooks, but just in case we glanced at that, and again, no difference there. Finally, we looked at the individual comments, the feedback from the students, and we got some great comments for how they're perceiving the class now. Assignments helped bring understanding to the materials. The module information was more than any other online class I've taken, which was very helpful and appreciated. And the modules offered plenty of material provided to help you learn. So in all the important measures as far as student satisfaction, we are seeing improvement with this gamification model. And so again, just to summarize what I've said, the DFW rates are remaining unchanged so far with gamification. We're trending the right way, but we'll have to keep an eye on that in the future. The students do perceive, though, that they are gaining knowledge and understanding of the course content. Again, I think that's an important thing to see in a non-majors course. Additionally, student satisfaction has increased, which again, you have students who are scared of science when they come in. Anything we can do to help them to not be quite so afraid, I think, is a bonus. For the future directions, we wanted to look at if the students are continuing to meet the learning outcomes for the course. I will tell you that at the time that I was aggregating all the data for this talk, we were being visited by SACs. And so let's just say that that particular department didn't really have a lot of free time to help me with my study. So that'll have to come in the future. And I just want to wrap up by thanking the many departments that helped me with this study. My learning designers, Colleen and Stephen, were absolutely amazing. that dating game was all Colleen's idea, and she just did an amazing job with it. I got data from our Department of Information Technology, Assessment and Institutional Research, research from Teaching and Learning Excellence. And I just want to point out, if you need any of my references, they're all attached as a supplemental document on my presentation. So you're welcome to look at that. But in the meantime, I'm happy to answer any questions that you might have.
Great job. Great job.
Thank you.
Great job, Cheryl. I really enjoyed that. And I think bringing a story to the way that we learn is going to become more and more necessary in the modern age. I think 10 years from now, how are textbooks, or how's that reading going to be? I think stories are a better learning method. And I think that's kind of what is happening in your.
Absolutely. Yeah.
It's great. So let's see a question we have here.
I do see one in our chat that, if I can, I'll read Gretchen's question. So Gretchen had asked about the different types of games that I've used or might want to implement besides the dating show. So for the sake of time, I couldn't show you all of the things that I've been using. One of my favorites that I've adapted, we have a game we play face-to-face, where the students are predators searching for a little crafting puffball prey in a giant piece of fabric. And we have like a red fabric with red and black puffballs, and then vice versa. And again, my learning designers adapted that one to be a game where they click on what they think are the prey in a very textured background. And so we can generate data about, so for the microevolution section of, are they able to hide from their predators? Are they not? How is that going to change that population over time? So we've done some fun things with that. And even some just mini games with, I also teach a science of cooking class based on Joe Provost's book, where we talk about you're having a dinner party and you have different dietary restrictions. How would you handle that? Which dishes could you serve and not? So I don't think it needs to be these big, elaborate things that the learning designers come up with, to just have a little bit of fun game and simulation in there.
Great job, Cheryl. I think to me these sound like stories, and and I do believe that is the future, that way that students aren't. We know that students aren't reading as much as they used to. No. Not reading at all. And I think this is a great strategy. So continued success in moving that forward in your publications in that. All right, so our last speaker is Kenneth Hallenbach from Reimagine Science. So can you introduce yourself and share your research with us, Kenneth?
Yeah, absolutely. I am excited to talk to you all today. Hard acts to follow, and actually I'm indebted to Cheryl for just going ahead and giving my introduction for me. Thank you. I'll have to do a lot less explaining of what gamification is. But let me introduce myself. I'm Ken Hallenbach. So I got my PhD at UCSF, and now I work at Genentech in South San Francisco. I'm a lab-based researcher, so I spend my time in lab trying to do early drug discovery research, or I shouldn't say trying. I do early drug discovery research, and so that's my day job. But when I'm not doing that, I'm really excited about different ways to address the kind of larger cultural challenges that science faces, both in society and within the scientific research culture. And so I've been working on some gamification, which you already all know what that is. So I'll just get straight into it. And again, as I've been saying, Cheryl did a great job explaining things, but having hands-on experience that's fun is really important to making labs seem less scary. So if you are a science student and you don't know what you're doing, actually getting your hands on a game, or, this is why we have lab-based courses as well. Basically everything we've been talking about, just making it fun, making it interesting. If we can connect that experience with role models that students connect with. So role models that look like you or that are from a culture you're from. These two things combined can make you seriously consider a STEM career, right? So if we wanted more Black scientists, having more Black scientists in the lab for students to see actually might be one of the tools. Well, it is one of the tools we need. Unfortunately, it's kind of this self-fulfilling prophecy, right? Where if a student doesn't see a role model or doesn't have a fun experience, then they might not choose a career outcome like this. And this is a problem that I've been thinking about a lot, right? Like, I grew up in an incredibly well-positioned situation from a scientific education point of view, right? But a lot of people don't. A lot of people don't have access to fun, engaging scientific education. And frankly, if you're interested in science, you're likely to meet a scientist who looks a lot like me. And that's not an accident. That's because that's what most scientists look like if you just took an average scientist in America today. So how do we address this fundamental challenge? It's not like the future has to be this way. And that's where I get really excited about the power of virtual worlds. So how does gamification fit into this? Cheryl just talked about it, and there's literally the same exact definition that she gave you. Hello, welcome. I'm back. I'll pick up here. I guess I had some stuff covering the top of my slides. So, you know, my slides are beautiful and I want you to see them. So we fixed that. So I'm going to briefly get into the gamification because I've done something a little bit non-traditional here, where what Cheryl was talking about is the standard form of gamification. So you take like lab research or your typical educational content, and you just add some elements of gamification. So like you can earn points and like maybe accomplish something, like you can cash those points in at the end of the semester. Or you could perhaps tell a story. And this is exactly what we were just talking about with the dating game, right? Is like, okay, like which of these different Neanderthal, Homo erectus, like who is it? They all have different parts of it, and that engages a different part of our brain. You can also do things like have achievements, like you know, who had the best meme? We all voted on it, right? So these are all things that you've seen so far today. And what I've done is I've basically taken this and ramped it all the way up to 100 and made an actual video game where it's more like the labification of a digital game than it is the gamification of an actual lab. And there are a bunch of reasons why I think this is powerful. One, it's pretty orthogonal. Like you're not going to just play a video game instead of go to school. So it's an additional tool rather than trying to replace the current content. I'm not a teacher, I'm a researcher. So I don't want to replace the teachers. I also think there's a lot of things the virtual world allows us to do that reality is we're constrained by, right? We're on a Zoom call right now. So that's one example. Access, right? So, you know, we didn't all have to fly to the same place to listen to these talks, which is super powerful, right? Like I'm going to go have lunch in my kitchen in 25 minutes. That means lots more people can access the same information. And that's a really powerful tool. Second is scalability. Real world lab space is always going to be limited. Like if you're at a small school, high school or middle school, you probably don't even have a lab. And getting a student into a lab and getting them excited about research so that they'll consider a career can be challenging, but a virtual lab has no limits. You know, if you have a computer, you have a phone, you can get into lab. And then lastly, I've hinted at this, but this is really where like my core interest lies, is we can, in a virtual world, short circuit the problem of representation by just creating the world we want our students to be living in the future, so they can experience it today. Like they can go into lab and representation rates can be appropriate because like we decide in a virtual world what the rules are. And that is, I think, an extremely powerful tool. And lastly, I'm also excited about the collateral benefits of just normalizing science and the research process as fun and creative and human and something that we all can do and maybe all are doing at home, right? It's the scientific method is not scary. It's just something that humans do. It's how we ended up with candlelit dinners because we wanted the roast fish, right? I mean, this is all something that we can connect people to. So without any more to do, I actually want to show you the game that we've made. And so there may not be sound, but go ahead and watch this trailer. Awesome. So I hope you enjoyed that as much as I did. This has been a year-long process for me. I'm a development and a design team of one, so it's definitely a passion project. But what I've tried to make here is the concept of a science game that's fun, that might be an educational tool as well. So it's maybe 5% of the way to something that would be used as a final product. But we want to inform the design. And the reason kind of I'm going public about it now before I go any further is, as I said, I'm not a teacher, and I really do want to inform the design of this game so that it would be useful to the people who should be using it. So we're part-time and we're having a lot of fun. We hope that you'll reach out to us, and I'll tell you how to contact us at the end. I'm also actually going to let you play it right now. Well, not right now because I'm still talking, but if you go to terraprime.org/researchrush, you can actually play that demo that I just showed you. And it might take a few minutes to load, and we recommend Chrome. And if you can't figure it out, email us. But you can actually go and play what we have. It's a concept game. And what I'm hoping it'll do is allow us to spark the imagination of our potential partners, people who might want to be working with us. And the reason I want to do that is kind of because I think it could go all kinds of places, right? I think we could actually walk students through lab experiments in a world like this, have them design their own lab experiments, build in content, or make it customizable to your lab course where you want to do something in real life lab, but then actually go do an experiment that you don't have the funding for or the instruments for in the digital lab, right? And this kind of back and forth, connecting the real and the virtual, I think that's the future, and how to get there is something I'm very interested in. And of course, there's always just like the let's play a game and have fun to inspire our students. That is probably where we'll start. And then the last thing I want to mention before I kind of wrap up is something that Cheryl hinted at, which is that a lot of research into gamification it hasn't been as conclusive as you might think. A lot of studies that show gamification in educational contexts are either too small, or they're not designed properly, or they just don't yield the result you might expect. And so I've been thinking, like, intuitively, even in this session today, you can already see that it makes sense that games would be helpful educational tools. Can we definitively show that? And can I design my game so that it is the ultimate tool for showing that, right? And so basically where I am today is I'm like 5% of the way through game design. I haven't done any thinking about how to design a research study using this, like A/B testing or anything like that. So what I want to start doing is take this 5% of the curve and start bending it, right? So start actually talking to educational researchers, which is why I'm here. So please reach out. Start talking to people throughout the course of this year to build in elements that will be useful for students, useful for teachers, and useful for researchers. And then ultimately actually start to run those studies alongside the game design. So we could do like a small scale pilot, kind of like we would do a clinical trial in therapeutic discovery, right? Where we run phase 1 to make sure that like the drug doesn't kill you, right? We like make sure the game is actually fun before we roll it out to a whole class or something like that, right? And then ultimately, I really want to actually analyze these data, publish them, and have an impact on the overall field of research and lab and education and gamification. And so that's where we're headed. Thank you so much for listening. I really appreciate the time. Sorry for the technical hiccup in the middle. If this is something you're excited about, please reach out to me. I'm at info@terraprime.org, or just you can follow us on Twitter. And I've finally put the password here. So if you've gone to that site, you've noticed you can't get in yet. So if you do want to actually play this build, please go to Research-Rush and just use the EXP Bio 2021 password to get in. And send us your thoughts. We really appreciate it. So thank you so much. And I'll take any questions from the other speakers or anyone who's listening. Thank you.
Great talk, Ken. Hollenbach, I appreciate that. I actually video a lot of my labs that go through every step. And I was actually thinking that could be incorporated into a video like this, and then students do a certain activity at these, and that could be a pre-lab assignment. I think there's a future for that.
Yeah, I love it. I mean, I think so. I'm glad you think so too. Yeah.
So is there a way that, so I send students to, previously I sent them to like Journal of Visual Experiments and other places, but what I noticed is that the tools and stuff or the spacing is a little bit different, and the students have a hard time like connecting, oh, this gel box is different than this one, and they lose a sense of that. I'm wondering if we could incorporate these games into the tools we have in different labs in the future. I mean, you're at the beginning stages, but to make them to where they're designed to our specific spaces and
yeah,
a real video.
Yeah, I mean, you know, I've actually had an even crazier thought of like, let's let the students make their own game, right? Like, I mean, these tools exist, you know, you can actually have them in the same way that like Gretchen had people download the MyNote or like go to the site and build their own concept map. You can say, okay, like we've at the beginning of the course set up all the digital versions of the actual equipment. Your homework until our next lab session is to go in and build the digital lab so you're ready, you know, and we could set that up. I mean, it's all possible if it would be useful. So yeah, it's a great idea.
Empower the students. That's give them the tools of that type of creativity. If it's easy enough for them to do, which it sounds like you're able to develop that. So hopefully we're able to test that in the coming years, or maybe even before the next year if we are able to contact you. I think you shared your contact information. Is that on the comment section?
Yeah, I'm not sure exactly how people watching at home can get to it, but just gonna look up my profile on the Cadmium site, I guess, and you'll find it there. Yeah.
So can we bring everybody back in? We'll bring in Cheryl Cozina, Gretchen Oya, Karen Resendes, and we have Ken Hollenbach. I think our Ed Markland's here as the ASBMB and staff, and if we could just come in and if we have any other comments we like to say. I think this has been a great session. I think there's connections between all of our talks and in learning, and I think it's taking that technology to the next level where we're becoming more visual. I think we need the audio now to come into like more audio learning, right? Where it joins a lot of our visual, our game strategies, our concept maps, the memes. That's how you get people excited, isn't it, Karen?
Yeah, you don't know how excited they are going to be to hear you guys love their memes. Like that is the number one thing. They're so super excited about you guys understanding what they're doing.
So where are we next year? Are we all going to travel to ASBMB next year, I hope, and maybe meet each other in person, and maybe we can put a poster together where we're connecting everything.
Sounds great.
That's the plan. Love it.
It was great seeing you all. Thanks for your talks.
Thanks, everyone.
Thank you, everyone.
Here, have a great summer.